wanted to start with thinking about what generally comes to mind when you visualize what a seagrass meadow looks like. I think typically people imagine these tropical subtidal meadows, probably much like this image, creating these beautiful, vast underwater carpets of seagrass. But in Hong Kong, the story is very different. Here, seagrass meadows exist as typically small, fragmented patches in the intertidal zone, surrounded by one of the most urbanized and densely populated cities in the world. These meadows are under constant pressure from industrial pollution, urban runoff, and coastal development. Yet, despite these challenges, they persist, playing a vital role in coastal ecology and blue carbon storage. And in fact, last year, when I attended the UN Ocean Decades Coastal Cities Conference held in Qingdao in China, I think this really highlighted the stark contrast in some of the challenges that ecosystems in remote or rural places face in comparison to those found in coastal cities. And I think for Hong Kong in particular, the reality of these challenges highlights the resilience of these ecosystems and the urgent need to protect them in these urbanized coastal areas. And this is what I wanted to discuss today, looking at what threat these ecosystems face in Hong Kong and southern China and how they persist despite these challenges, and how we are implementing solutions to protect and restore these really vital ecosystems. Now, this story actually really begins with the rapid urbanization of Hong Kong from its origins as an archipelago of small fishing villages. Hong Kong has transformed into a global financial hub known for its towering skyscrapers and these bustling ports. However, this transformation of urban growth has really come at a cost. And we've had over 7,000 hectares of land reclaimed since the 19th century. And while this has created space for infrastructure and housing, it's also drastically altered the coastline and squeezed these intertidal habitats into fragmented patches. But there's this weird paradox that despite this intense urbanization, Hong Kong remains a place of actually really ecological. So with 7.5 million residents packed into this small cluster of just over 260 islands, it's actually the fourth most densely populated region in the world. Yet its subtropical waters that are characterized by hot and wet summers and mild and dry winters still support vibrant marine ecosystems, including coral reefs, mangroves, oyster reefs, and of course, seagrasses. And these intertidal meadows are vital habitats for species, including the endangered Chinese horseshoe crab. And this just really proves that even in one of the world's most busiest cities, that nature persists. So what species are found here? The dominant species in Hong Kong are Holophila ovalis and Zostera japonica, which is so rare here that Hong Kong's 100 rare plant species list. And there are also only a handful of populations of Holophila beccari, which is a species classified as vulnerable on the IUCN red list. There are also records, historical records of Rupia maritima and Holophila minor, though those haven't been confirmed recently. And if you're wondering what these characters are in the top right, that is the Cantonese symbols for seagrass, which is hoi chow, and that's seagrass in Cantonese. So Hong Kong's dense urban environment and ongoing coastal development have placed immense pressure on seagrass meadows, and this just really compounds the challenges these ecosystems must overcome. In many ways, the challenges that they face here mirror those of coastal ecosystems globally. And this kind of makes Hong Kong a microcosm of global seagrass decline. So, for example, land reclamation, as I've mentioned, also leads to high sediment loading, and this is also then often linked with water pollution, from industrial discharges, agricultural runoff, and urban wastewater. And then this goes on to introduce heavy metals and excess nutrients, making eutrophication events really common, particularly during the summer months. At more local scales, however, seagrasses face impacts from extractive fishing practices. And this includes things like mussel digging, which is really a common practice throughout the intertidal mudflats of Hong Kong. But large scale, we're increasingly seeing the impacts of climate change in the region, with rising sea levels, warming waters, and extreme weather events significantly threatening these ecosystems. And in fact, this year alone, Hong Kong has been hit by 5 tropical cyclones with wind speeds reaching up to 150 kilometers an hour. And we've also experienced 5 black rainstorms, including a record-breaking event in August this year, which saw over 358 millimeters of rainfall in a single day. And this is the heaviest daily rainfall since 1884. And then this leads to widespread flooding and landslides. Yet, despite the ecological importance and the ecosystem services they provide, we've had very little data on how Hong Kong seagrasses are responding to these pressures. So, for example, how much have they declined? Where are the population refuges or climate resilient genotypes? What's the tipping point? And like in so many other places globally, these knowledge gaps present a significant barrier to effective conservation and management. So this is where I would like to introduce our team. So led by Associate Professor J.D. Gatin-Espadilla, or we just call him JD, our team brings together really diverse expertise. So this bridges seagrass ecology, physiology, genetics, and blue carbon science. And over the last few years, we've built collaborations with universities, NGOs, and government agencies, and this allows us to directly inform management and restoration throughout Hong Kong. JD also formed the Hong Kong Seagrass Restoration Association to promote the regeneration of seagrass meadows in Hong Kong through transplantation as well as public education. So, as I've just mentioned, one of the most urgent gaps we're tackling is distribution data, because you can't protect what you don't know exists. The last comprehensive published study on seagrass distribution was actually conducted in 2019, but species assemblage and area cover data were not made publicly available. So this leaves 5 years of undocumented change in what are highly dynamic populations, and this leaves management efforts relying on outdated information. But our current research is changing that. So by combining historical records and habitat suitability modeling, we've already uncovered new populations and unfortunately documented losses where seagrasses have disappeared. And through this extensive modeling, we've identified several sites that show promise for restoration, particularly in sheltered bays with historical seagrass presence and where adjacent habitats like mangroves or oyster reefs could provide natural buffers against things like wave energy. Critically, as the only Asian partner in the Smithsonian's Global MarineGEO Seagrass Working Group, we're not just collecting monitoring data, we're also recently, we helped to refine how seagrass monitoring is done across partner sites globally. But monitoring distribution is really only just one small part of the puzzle here. And to conserve these meadows, we must understand whether their ecological function degrades under all these pressures, particularly from things like eutrophication. And our stable isotope work exposes the hidden cascades of nutrient provision. So this research quantified the effects of seasonal variability and anthropogenic eutrophication on seagrass-associated biodiversity and trophic dynamics. We aim to test the hypothesis that nutrient enrichment alters primary producer abundance, And thereby this restructuring of macrofaunal trophic interactions, potentially with seasonal mediation via temperature fluctuations or freshwater inputs. So to assess these dynamics, we analysed carbon and nitrogen stable isotopes in 280 macrofaunal samples, including crabs, bivalves, and gastropods, and these were collected from 2 seagrass populations with contrasting nitrogen enrichment levels. So in the high nitrogen sites, they exhibited reduced species composition and richness, and this indicates trophic homogenization under eutrophication. In addition to this, consumer trophic positions also shifted, with algae displacing seagrass as the dominant carbon source. In the wet season, nitrogen pulses exacerbated niche compression, and we think this is linked to freshwater-driven nutrient loading. So in essence, these results demonstrate that eutrophication not only directly stresses seagrass, but also decouples their role as foundational carbon and nitrogen cyclers, which is really a critical concern for Hong Kong and other urbanized estuaries. And quantifying these thresholds, such as nitrogen loading, limits and niche collapse points, is really essential for managing seagrass ecosystems under all these escalating anthropogenic pressures. What's more, we also explored how chemicals found in sunscreen may be acting as an emerging threat to seagrass physiology. So several studies have noted UV filters and sunscreen have varying degrees of impact and toxicity on different ecological receptors, the classic case being in coral reefs. But in terms of seagrasses, exposure can impair seagrass photosynthetic efficiency, and this works by damaging internal structures and metabolic processes, and potentially reduces population resilience. So through a controlled microplate experiment using Halophila ovalis, we tested organic versus inorganic sunscreens with reef-safe and non-reef-safe claims. And these were tested at different concentrations over with a 72-hour exposure period. So the key findings using chlorophyll fluorescence and spectrophotometric analyses showed significant reductions in FBFM and chlorophyll content with organic non-reef-safe sunscreens. So this indicates photosystem 2 damage. And I think what's really notable here is the so-called reef-safe sunscreens showed comparable impacts to the conventional non-reef-safe formulations. And this potentially highlights the lack of standardized ecotoxicological criteria for these labels. The observed non-photochemical quenching increase suggests sunscreen exposure triggers photoprotective mechanisms, while chlorophyll degradation in organic treatments may reflect incidental nutrient toxicity. And overall, these results demonstrate that even short-term exposure, this was only 72 hours, alters seagrass photophysiology and establishes sunscreen pollution as a measurable stressor in urbanized coastal ecosystems. So as I've mentioned, nutrient pollution and urban runoff are really key threats in coastal ecosystems, and particularly in Hong Kong. And previous studies have demonstrated that nutrient inputs can also disrupt sediment carbon sequestration processes, as well as carbon storage capacity. So our study aimed to quantify how seasonal eutrophication impacts seagrass carbon stocks and the potential for these overlooked blue carbon sinks to be used in climate mitigation. A few of the key results we found are that seasonality and local nutrient loading are driving changes in blue carbon stocks, with high nitrogen levels from agricultural runoff and urban wastewater really detected in our stable isotope analyses, particularly during the wet season. But label carbon from algal overgrowth really presents a key risk here for the rapid remineralization of this stored carbon. So the carbon here is just, it's more at risk and it's not as stable. We also uncovered some species-specific responses between the seagrasses, with Halophila beccari showing higher below-ground biomass carbon storage than Halophila ovalis. And we think this is potentially due to their dense rhizome structure trapping particles. The mean sediment carbon stocks of both species in the upper surface sediment were comparable with mean sediment carbon stocks reported for seagrasses in Southeast Asia, as well as globally, as you'll see on this slide. And lastly, something else that we wanted to explore was estimating the social cost of carbon and the carbon dioxide equivalent of these carbon stocks in Hong Kong, considering that these are small fragmented populations. So we utilized a regional approach to estimate the social cost of carbon. And we did this instead of a global approach, as country level estimates promote a better understanding of regional impacts of carbon emissions. And this leads to improved adaptation and compensation measures generally. So Hong Kong seagrass meadows store between approximately 780 and 972 micrograms of carbon dioxide equivalent. And this equates to a value of 300,000 Hong Kong dollars in social cost of carbon and climate benefits. And while this is significant, it should also be noted that these estimates were only based on the upper sedimentary layers, so only the top 15 centimeters of the surface sediment, and also only investigated 2 sites. So only 2 sites were surveyed, representing just over 16 hectares of Hong Kong's total coastal area. So not including many of our other sites, and not including the whole of Hong Kong's coastal area, which is approximately 164,000 hectares. So 16 hectares to 164,000 hectares is, a really big gap in terms of what we've actually accounted for here. So we really need further annual assessments of total carbon stocks for the entire extent of Hong Kong seagrass meadows, or to model this, and that's really essential to comprehensively determine the blue carbon potential of seagrass ecosystems throughout Hong Kong. And this work highlights that we really must protect meadows from nutrient overloads to safeguard their role, particularly if we want to include them in Hong Kong's Climate Action Plan for 2050. So in terms of policy, the carbon sequestration potential of these seagrasses have really important implications for carbon accounting in Hong Kong, particularly if they were begin to become included in national carbon inventories or voluntary carbon trading schemes. And this would really enable us to unlock blue carbon financing for our restoration work. And as you see here, this work was recently published in Estuarine Coastal and Shelf Science as part of a special issue on blue carbon monitoring, assessment and restoration. So if you want to read that paper, then please scan the QR code and it'll take you there. So next we investigated carbon storage in 2 critical intertidal seagrass ecosystems. So we looked at Paknai, which is known for its extensive oyster agriculture and supports really high diversity of mangrove and salt marsh species, as well as the vulnerable Halophila beccari. And then we also looked at Sandtow, which is another ecologically significant site, hosting a mixed species seagrass meadow of Halophila ovalis and Zostera japonica. So sediment cores were taken only in the top 3 centimeter depth and were collected along high and low shore transects during the dry winter and wet summer seasons. What our results showed is that there are clear site-specific carbon dynamics. So at Sandtow, high shore carbon content increased significantly in the summer, exceeding low shore values, whereas in Paknai, the inverse trend was found with high shore carbon content decreasing in summer, while low shore winter samples showed actually the highest variability. So the potential drivers of this site-specific variability include local hydrodynamics. So at Sandtow, there's some fresh water input, and this likely enhanced organic matter retention. What's more, species differences in seagrass assemblage, such as Halophila bakari, its proximity to mangroves, may also drive a Paknai's unique carbon patterns. So the implications of these findings are important when developing monitoring frameworks, with shore level and seasonal sampling both critical for determining accurate blue carbon assessments. And in a local context, this work again can also feed into management action by integrating sediment carbon data into Hong Kong's Climate Action Plan for 2050. From the extensive monitoring that I've mentioned, we identified these recovery or restoration hotspots. And we identified these as having potential for targeted interventions, such as piloting small-scale transplantation to potentially bring back these lost meadows. And early experiments at these sites have already shown really encouraging seedling survival rates, and this offers us a potential roadmap for scaling up these efforts across the region. In fact, in August 2023, the team implemented a rhizome transplantation technique adapted from a research group in Qingdao, China, and this focused on restoring seagrass in Seokyu-1 on Lam Island. And this methodology prioritized using long rhizome fragments to enhance establishment potential. And as you'll see here, this is what the site looked like originally. And then through quantitative validation, using remote sensing, our drone footage here, which was taken only a few weeks ago, reveals seagrass dramatic expansion in area cover over the past 2 years from what was just originally a few rhizome fragments. And as this is Hong Kong's first documented successful restoration attempt, these results validate our site selection criteria, which are sediment stability, moderate hydrodynamic energy, and freshwater inflow. So we're now looking at how we can map the path forward to scale this success at other sites. And one potential solution to this is seagrass mariculture. So this isn't new, but by optimizing techniques for growing and transplanting seagrass, we can restore degraded meadows and enhance their carbon storage potential. Using our indoor seagrass nursery at the Swire Institute of Marine Science, or SWIMS, we employ an open flow-through system, maintaining all 3 species under controlled conditions. So as I mentioned, we are currently optimizing our mariculture protocol, as we have seen evidence of some light limitation on the plants, as well as we want to experiment with artificial sediment to reduce the impacts on donor meadows, particularly if we want to scale this, and also investigating fertilizer addition into the sediment to help promote faster recovery and growth post-transplantation. So using a factorial experimental design aimed to quantify the effects of light, and addition of fertilizer on the shoot number of these 3 species. So over the course of 12 weeks worth of monitoring, the number of shoots in the culture tank increased for all species, with Zostera japonica showing 11.2 times shoot proliferation. And the coverage of Halophila ovalis also increased by up to 122%, with reproductive structures only actually observed at high light intensities. Then in a separate study, we wanted to investigate the use of different plant material, as obviously collecting shoots instead of cores, again, would help reduce the impacts on donor meadows. So we also investigated the use of open versus closed systems with the aim of minimizing algal competition, which is a really common issue for seagrass mariculture. And over, again, these 12 weeks, no significant interaction was found at the end of the experiment between starting material and flow system on shoot density or photosynthetic performance. However, the closed system significantly increased shoot density, as you can see here. So to scale, we're now partnering with the Ocean Park Conservation Foundation, and we want to be able to upscale our nursery capacity for restoration, and this would allow us to manipulate different factors to optimize our mariculture protocol. And this is especially important as there are no publications on seagrass mariculture for the seagrass species that we have. So the team has also been engaging with the Global Seagrass Nursery Network to share what we've learned, including the mistakes and, of course, all the many challenges we've had along the way. And yeah, as I mentioned, we can share this with the Global Seagrass Nursery community. And in parallel to this, we're also investigating the potential of in vitro micropropagation. And this method would also further help reduce, again, impacts on donor meadows. And we have started by investigating nitrogen preferences through the addition of nitrate and ammonium at different concentrations. And we found that after 7 days, nitrogen concentration, particularly for nitrate, was found to be a significant negative predictor for photosynthetic performance, particularly at high concentrations of 1,000 micrograms per litre. So here the observed decline in photosynthetic efficiency at these higher nitrogen concentrations suggests nitrogen toxicity. And this phenomenon has also been reported in other seagrasses, finding that an excess of nitrogen causes tissue damage by disrupting nitrogen metabolism, and a decline in photosynthetic efficiency due to disrupting photosystems 2's ability to conserve energy from absorbed photons. The decreased photosynthetic activity further leads to a reduction in energy uptake and biomass formation, ultimately inhibiting plant growth. And these significant declines in photosynthetic performance under high nitrogen exposure suggest that Halophila ovalis is actually really susceptible to eutrophic conditions, as some of our other research has highlighted, which is becoming a more prevalent threat in Hong Kong's coastal regions. But restoration isn't just about the science, it's also really about the people, and that's why we've partnered with WWF to bring the public into our work through hands-on transplantation events. So together with local volunteers, we've outplanted hundreds of Halophila ovalis seedlings, and by utilizing social media and these face-to-face interactions, we're turning participants into ambassadors for seagrass conservation. And this groundswell of local action is now gaining institutional recognition, with the University of Hong Kong recently featuring our research as part of a social media series that was highlighting seagrasses as a nature-based climate solution, one that delivers environmental, social, and governance benefits as part of ESG. So meanwhile, the Hong Kong Maritime Museum has also helped us reach new audiences, showcasing our seagrass research in one of their new permanent exhibits. And this partnership proves that when science meets storytelling, we can also help shift perceptions of seagrasses from these invisible meadows to recognized climate solutions. But scaling up this work requires overcoming some really difficult challenges. And a few of the key hurdles, as well as opportunities that we're navigating, include nursery maintenance, which is really demanding and requires ongoing manpower, but we don't really think that there's too many ways of getting around that, as everything that can be automated is already. Additionally, remote sensing in turbid Hong Kong waters remains technically really difficult and quite challenging. But there's also some key opportunities at hand here, including continuing to build new partnerships with NGOs to scale our seagrass nurseries citywide and have them more public facing, and potentially tapping into voluntary carbon markets to unlock additional funding for our restoration work. And in fact, one new project that we have just got funding for is a novel restoration project using abandoned salt pans as outdoor seagrass nurseries. So in Tai-o on Lantau Island, there is a history of salt production, long-running history of salt production, and in the 1940s, the production amounted to approximately 25,000 pickles. And a pickle is a traditional Asian unit of weight, defined as much as a man can carry on a shoulder pole. But in this case, that is equivalent to 1,512 metric tons, which is significant. But these historic salt pans provide, they're fantastic because they provide natural protection from strong wave action and currents, and this is ideal for seagrass establishment. And these salt pans also have tidal exchange, which will help avoid eutrophication while maintaining seagrass productivity. So the next stage of this project is to assess climate resilience and determine the vulnerability of these salt pans to extreme rainfall and the associated freshwater influx, as well as rising temperatures or temperature variability. For this project, we also are going to partner with local fishers and schools to help co-manage the ponds and raise awareness of seagrasses globally. So this isn't just problem solving, it's also systems building. And this type of systems thinking is really a non-negotiable, particularly as Hong Kong seagrasses don't exist in isolation. They're part of a much larger interconnected system. And as a keynote in the Greater Bay Area, Hong Kong sits within one of the world's most dynamic economic zones where rapid development meets the vast Pearl River estuary. And here, heavy sediment and nutrient loads create a quite unique salinity gradient along the western coastlines. And this shapes estuarine conditions that then go on to influence seagrass survival. However, effective conservation also requires robust policy and governance across borders, and our research has found some really stark mismatches in management policies across these borders within the GBA. And the regional challenge here demands coordinated science and governance. So how do we bridge this gap? Our team is working to help build stronger regulations, climate action plans that include seagrass protection, and critically, we're helping to promote regional data sharing. So this brings us to the roadmap ahead. So our research priorities target both knowledge gaps as well as key action gaps. So we're deploying cutting-edge tools by expanding standardized monitoring to predict climate refuges and harness predictive models to identify climate resilient genotypes. We're also using eDNA to detect hidden populations of seagrass, as well as obviously, the communities they support. We're in protections, and we're working with government agencies to conduct IUCN red list assessments for both species as well as ecosystems. And all of this while scaling restoration. And we're particularly interested in using these abandoned salt pans as outdoor nurseries to try to prime seagrasses for real world conditions and hopefully improve outplanting success. So the story of Hong Kong's seagrass ecosystems mirrors the global paradox of the Anthropocene. These high value blue carbon sinks are declining at an estimated rate of around 7% annually, precisely when their climate mitigation and coastal resilience services are really actually at the most critical. Yet all this research that we've conducted demonstrates that even hyper-urbanized coasts can be resilient to global change and be restoration frontiers. So the call is actually really clear. As scientists, we must continue to work together with policymakers, NGOs, and the public to safeguard these vital ecosystems and accelerate restoration targets. So none of this happens without collaboration, and I'd like to thank our collaborators, including WWF and the Ocean Park Conservation Foundation, who I've mentioned during this presentation, and of course, several of our other funders, including the Environmental and Conservation Fund and the Lantau Conservation Fund. So thank you for your attention, and I'd now like to welcome any questions.
Thanks, Katie, for your presentation. Do we have any questions for Katie?
Thank you for your presentation, Katie. Was amazing. My name is Lisa, so I'm from Mauritius, and I wanted to ask you more about the restoration. Was it by seedling or was it shoot transplant?
It was shoot transplant. We have found seeds and inflorescences in the lab, but it's just not really practical to be able to scale this because they're so small, the inflorescences, that it just takes too much time to go through and sort these. It's not like with Zostera marina, you can grab handfuls of shoots and then sort through them quite quickly. So they're producing very few seeds, they're really small. It's just not practical, which is why we've gone through this seagrass mariculture route and also exploring these outdoor nurseries as well to be able to scale that in a meaningful way. Thank you.
And if I may ask, what are the method that you use for the shoot transplant? Because we were also doing some literature review on restoration, and we saw there was a plug method, there was a staple method. So which one worked best for you, and which species did you use for the restoration?
Yes, so we used Halophila ovalis, as this is the most abundant and widespread species throughout Hong Kong, and this also allows us to some of the populations of the other species that we have, if we were to collect donor material, we would essentially wipe out those populations. They're so small. So yes, Halophila ovalis is really important for us, and that's also, again, why we have the nursery, to be able to have backup material for a lot of our restoration work. And then in terms of methods that we've used, so we, as I mentioned, for SOCU1 on Lama Island, we used these long fragments. So we didn't cores, we kind of dig them up, kind of using your hands actually, so it's more delicate. And then you can kind of take these long rhizome fragments and just plant those. We don't anchor them or anything like that. Actually, some of my PhD work from back in South Africa found that by anchoring them, yeah, it's not as preferable as using cores. And in Hong Kong, we've also used cores, and we're not sure whether it was, I think it was the sites that we selected, but cores have not found to be very successful here. We think as well that's also because the method of taking a core causes too much damage to the rhizome fragments, whereas if you can gently dig up longer fragments, then there's less post-transplantation stress on the plant, and this allows it just to, particularly if you have the growth tip, this then just allows it to laterally spread and grow. But please feel free to send me an email if you have any specific things that you want to work out.
Okay, thank you so much.
I also have a few questions.
Yeah.
I'm just gonna build on from what Lisa is asking. Again, thanks for your presentation. It was really interesting, and it's also very coming from Singapore, it feels very similar. We've got the same problems, land reclamation, all of the above. We are starting a restoration project in Singapore. So I think it would be great to be able to talk to you offline a bit more about it as well, because we are also trying out Halophyllus. With your long shoots, how long did that take? Like all of it, like preparation, like digging it up and all that, and then having to plant it, because it looks like you just like pushed it into the sediment, didn't you?
Yes, yeah, we just pushed it into the sediment. So we actually didn't take very many fragments that time. So I think we took about a dozen. So yeah, the preparation and planting, it was 2 days worth of work, and it would have taken less if we could get from between sites quicker with the tides. So yeah, a couple of hours at each site really. But if you're closer to each site, then you could probably do it in the same day especially if you have a spring tide.
So you mean just with those dozens of shoots that you took, it became that entire meadow that you showed?
Yeah, which is
Wow.
incredible. Yeah, it's incredible. Yeah.
Oh, that is really amazing.
Yeah.
Congratulations.
Thank you.
You mentioned
I can't actually really take any credit for it because it was
okay. Congratulations to your colleagues.
Yes, I know. As I say, it's a team effort. So yeah, I just want to highlight some of the work that they've been doing as well. Yeah.
Oh, that's really amazing though. You mentioned though in that you think that maybe it might have been the site suitability of that area as well, and that one of it is potentially freshwater inflow. Why do you think freshwater inflow was advantageous, or did I get that wrong?
Yeah, it's counterintuitive based on what I expected. But all of our most successful seagrass sites throughout Hong Kong have a small degree of freshwater inflow. We're not really sure what is kind of driving this. The best we have at the moment is that it helps prevent some desiccation during low tide, and potentially increases the macrofaunal diversity that reduces algal competition at sites. Yeah, we're interested to explore that.
Good. So most of your sites that you think are suitable have some fresh water.
Yeah.
Interesting. And it's good to hear that you guys are in touch with the Global Seagrass Nursery Network, because yeah, that's way not enough tropical information in there.
Yeah, 100%. And I think, like most of the seagrass work, there's a strong bias towards
Zostera marina.
Europe and North America and the species that are found there. So you kind of have to start with that really foundational understanding of growth preferences, and then how you can optimize that from zero, really. Yeah, because it's different for every species globally. I think there's some common rules of thumb, but yeah, there's definitely things that we've found can optimize our mariculture protocol, and testing different things has really helped, and that's why teaming up with the Ocean Park Conservation Foundation has been really helpful, because Ocean Park is a marine, it's basically an aquarium in Hong Kong, but it's also a theme park. So they have tons of aquaculture experience, and they also have incredible large tanks and facilities to be able to test a million different variables to optimize what we're doing, which is really exciting.
Oh, that's good. Lisa, you've got a question?
Yeah, I wanted to know, are there any challenges that you encountered when you did your restoration in terms of like growth of algae, or like any kind of challenges? And also, I wanted to know about your ecosystem function. I think you mentioned eDNA, so I wanted to know like what is the period of time that you started seeing the first observation of fish coming to a new resting site? Because normally they are attracted when there is something new in the lagoon. So in terms of eDNA or like invertebrate analysis that you did among the monitoring.
Yeah, so some of the challenges that we've had with our restoration work is definitely identifying suitable habitat to restore. So as I say, we have used habitat suitability modeling, although the data available is quite coarse scale. So we've actually found that we've actually been relying more on historical records to be able to potentially identify where new seagrass meadows are, as well as where seagrasses have historically been found, to then pursue restoration, or some of the sites that that would otherwise be very suitable have a high degree of direct anthropogenic impacts, like I've mentioned with mussel digging. So any restoration that we did there would probably be in vain, unfortunately. We are also later this year going to be working within a marine park to do some restoration, and we think that the site had historical seagrass presence, and being a marine park, this will hopefully fit two of the key criteria that we need to overcome. And then, yeah, as I mentioned, with the restoration and having our seagrass nurseries, manpower is a massive challenge that we continue to face. We're all, the team is all really busy, finishing PhDs, conducting other research. There's a million responsibilities that we have individually, and then we have to constantly be working on cleaning the tanks and reducing algal competition. So yeah, that's really difficult. And then the other question you had was, sorry, the line was a little faint, but I think you were asking about when fish returned to seagrass meadows once they had been restored. So we haven't explored using eDNA to look at seagrass community yet. So this is another project that is in the pipeline that we're really looking forward to and excited about. But we have used eDNA. We have conducted a study that I didn't have time to kind of just talk about in my presentation, where we're looking at being able to sample sediment and water to potentially be able to find seagrass meadows in sites where the habitat suitability modeling or historical records have not previously identified. Because another challenge that we have in Hong Kong is that we think that the seagrasses here are facultative annuals, which means that they kind of disappear over the winter typically, but then can return during the summer. So it's not that they've died off, it's just that the rhizome network is in the sediment. So if we can use eDNA, then we can potentially continue to conduct these surveys throughout the winter to check for seagrass presence, absence, because as I mentioned, Hong Kong is an archipelago of 260 islands with equally with an infinite amount of little bays and estuaries and things, complex hydrodynamics. So if we can use the DNA to potentially help us narrow that search down for new sites or existing sites, then that would be fantastic for us. Yeah.
I have 2 more questions and I'll let you go. With your nursery, have you outplanted any of them? Have you tried? Have you outplanted?
Yes, we have. And yeah, we're finding really good success with that, actually. We want to be able to move from just growing them in a nursery to using these outdoor nurseries on the Tayo salt pans. And that's because there's a lot of literature showing that stress priming of plants enhances their ability to withstand stresses in the future. And we kind of want to harness that work and put it into practice.
Yeah. Yeah, cool. And you, with your outplanting, it has also been successful. So you've just moved it like this, I'm assuming, in the course.
Yeah, exactly. So we take some initial material, bring it back to the swims facility, get it to grow. So we just have like a small amount of seagrass and then put it in a large container. Once the container's full, then we can transplant back out again.
Cool. And also, sorry, so lastly, you mentioned potentially the carbon market for funding any kind of future research. Do you know broadly, is there much interest in that in Hong Kong? Like, because we know there is interest in, for example, Posidonia, but then our tropical seagrasses don't really have this giant mat, right? So it's not like our carbon stocks are that high. It's not mangroves. Do you know if there's interest in that in Hong Kong? Is that?
Yeah, it's an interesting question. I think, with Hong Kong being such a financial hub, there's interest in anything that can potentially do money. So at the moment, carbon trading I think is still quite difficult, even for areas that do have these really expansive meadows that could potentially generate more significantly more carbon stocks than here in Hong Kong. But that is, I think voluntary carbon trading schemes are a good alternative to that for the interim. the challenge there is around who owns the seagrass, right? Like our lab can't really sell the seagrass because we don't own it. So I think there's still a lot of legalities that need to be ironed out. I think for us at the moment, the key interest in our seagrass restoration work has come through ESG, so environmental social governance, because in Hong Kong, companies receive large tax breaks for doing this, and it's also actually required from the government if you're a company of a certain size or a certain income level. So yeah, they've helped fund some of our mariculture work, and they've helped fund that work has also helped fund us getting a research assistant for the lab, and also helps fund some of our outreach activities in schools to be able to educate learners on what seagrasses are, why they're under threat, things like that. So it helps us really engage with the local community, actually.
Oh, wow, that's really good to hear that there's that alternative source of funding. Alrighty. Lisa, do you have any more questions? If not, we'll let Katie go. Yes, Lisa has
I
questions.
just don't want I just wanted to know more about the in vitro propagation for seagrass. Is that what you mentioned? If I could get some more information on that and how that's helping in like restoration.
Sorry, could you repeat that? The line's a bit quiet.
So if you could like have more information on the in vitro propagation of seagrass. Is that used for your nursery?
Sorry, I can't quite hear the question.
Yeah, sorry, Lisa, your line's a bit thinny. Maybe if you talk slightly slower and louder.
I wanted to know more about the in vitro propagation and your nursery for the seagrass. Is it also like what you're using for restoration purposes?
So for the in vitro propagation, micropropagation, we are not at the stage yet where we are wanting to use that for restoration. Mostly because we want to run more tests and experiments to kind of understand. We're also using it as a method of understanding nutrient preferences for the seagrass species that we have. And as an aside, we also want to then look at, because we wouldn't really want to put the agar into the seagrass meadow. I know places where they are doing that, but I think we're a bit worried about unintended consequences. So we want to kind of study the impact of taking the seagrass out of the agar and then putting into sediment in a controlled system first to see how that works. So I think we're a little bit away from using in vitro micropropagation for restoration, but I think it is proving to be really useful to understand nutrient preferences and nutrient concentrations for the seagrass species that we have, which facilitates our seagrass nursery work.
Thank you, Katie. Amazing presentation.
Thank you
The
so much.
restoration.
Thanks. And yeah, as I say, feel free to contact me if you want any more advice on seagrass restoration.